rest of the traits we're going to talk about in this chapter are traits that don't quite fit Mendel's principles. Remember, he thought everything was just dominant or recessive, and that worked in the pea plants. But what we've discovered is that not all genes work that way. Not every trait's determined by just one pair of genes. Not every trait is clearly dominant or recessive. And so that's what I'm going to talk to you about in the remainder of this chapter. The Punnett squares and product rule will still apply. You'll still be able to use it. You're not going to learn a new kind of cross. What's really going to change are the phenotypes, the outcomes of the crosses. So you'll see what I mean as we go. So the first thing we're going to talk about is what's called incomplete dominance. So incomplete dominance is exactly what it sounds like. It's where neither one of the alleles is completely dominant or completely covers up the other one. And so when you cross these two alleles, the heterozygous one is an in-between phenotype. And the textbook example that every book seems to use for this is what's called the Japanese 4 o'clock plant. So when you cross a red one with a white one, the offspring come out pink, an in-between color. Now we know that the reason that this actually happens is because if you think of the genes as big R and little r, big R codes for making red pigment. And little r basically codes for making nothing. So if you're big R, little r, you make a certain amount of red pigment. But if you're big R, you basically make twice the amount of red pigment. And so this would end up being red, whereas this one with half the amount of red pigment ends up being pink. You may wonder, why aren't all traits like that? I mean, why isn't being heterozygous in the pea plants give them lavender flowers or make them medium height instead of tall and short? And it just turns out that in some genes, having one copy is enough that you can't distinguish a difference. So bottom line, if you see a trait with incomplete dominance, you're going to do the same kind of Punnett squares, but one thing you may notice is that, especially if you see this on a test, they may use superscripts or subscripts instead of a capital letter and a lowercase letter. Since sort of having a capital letter kind of implies that the capital is dominant over the lowercase, in a lot of cases for these traits, you might see something like this. C1, C1 for red, C2, C2 for white, and then C1, C2 for pink, kind of so that neither one of them really is dominant over the other. To be honest with you, when I do these crosses, I still just use big R and little r, and you're welcome to do that. The only thing I would warn you about is remember, as you're doing the cross, that you've got to keep in mind that big R, little r is not going to be red. It's going to be pink. It's going to be an in-between color. If you forget that you've assigned it that way, you might make a mistake. And this is just a picture showing the cross red times white. You get pink, but when you cross the pinks, you'll see that you can actually get red, white, and pink. We're actually going to do this cross in a moment. Now, very similar to incomplete dominance is something called codominance. So in codominance, instead of getting an in-between color, you see both phenotypes at one time. And I always get people that get a little confused on this, but it's really not that confusing. So a roan cow is what happens when, this is a roan cow right here, it is what happens when you cross what we would call a red cow, which is really kind of this rust color, with a white cow. Notice you don't get pink. What you get are blotches of red, so red, one of the phenotypes still shows, and white, the other possible phenotype, still shows. So you're actually seeing both phenotypes at the same time. So if you cross black and white and you get gray, that would be like incomplete dominance. If you crossed black and white and you get black and white spots or blotches, then that would be codominance. And another example of it is what we see with human blood types. It turns out that blood type A and B are codominant. Blood types are going to come up later because they're an example of something else as well. But if your blood type is A, you basically have a protein called A on your red blood cells, which is you see in the picture. If your blood type is B, you have a different protein called B. If your blood type is AB, which sort of got cut off at the bottom here, you don't have an in-between shape. You literally have A and B. You have both of those proteins. So that's the difference between incomplete dominance and codominance. Now, what does a Punnett square look like for this? Well, here are a couple of practice problems I'm going to run through very quickly. So it says to cross a red flower with a pink flower. And notice they preface this by telling you the trait shows incomplete dominance and that red times white makes pink. So that means our pink flower is going to be, I'm going to still use R's. I'm going to make it big R, little r. And my red flower would be big R, big R. Even if they didn't say homozygous, I could just say red. And I would expect you to know it has to be big R, big R. So be very careful, because I get people on the test that will come to me and they'll say, oh, it says to cross a red with a pink, but how do I know if the red is homozygous or heterozygous? And I won't be able to help you. But you're supposed to know because they're telling you it's incomplete dominance, which means that red, pink, and white, all three genotypes look different. Big R is red. Big R, little r isn't red. It's pink. So if I say a red flower, it has to be big R. And then little r, we're using for white. So if we cross a red with a pink, it would look like this. Big R, little r is my pink one, and my red one is big R, but I only need to write that one time. Remember, we can still do that. My genotype ratio would be half of them big R, and the other half of them big R, little r, and my phenotype ratio would be that half of them would be red, and half of them would be pink. Same Punnett square we've been doing, but your interpretation of the results is changed because of the fact that this is incomplete dominance. My second one, we're crossing two pink flowers. So that has to be big R, little r with big R, little r. Again, notice this is the same Punnett square we've done multiple times, but our interpretation of the results is going to be different. If this was a regular Mendel cross, you would get 3/4 red, 1/4 white. But this isn't. This is incomplete dominance. So we're going to get 1/4 big R, red, 1/2 big R, little r, those are pink, and 1/4 little r, and those are white. So that gives you two examples of incomplete dominance. Codominance would work the same way. All right, another thing that you might see, and this word is not going to appear on the AP exam, but I have seen traits like this in the AP exam, is something called epistasis. Epistasis is where there's more than one gene that codes for a trait, and the combination of two or more genes gives you different colors. So in this case, we're talking about black, brown, and yellow labs, Labrador retrievers, dogs. And notice it's two genes. There's a gene B and there's a gene E. Well, it turns out that if you have big B anything, big E anything, you will be a black lab. But if you have the specific gene combination of anything, big B or small b, with little e, you'll be a yellow lab. And if you specifically have the combination of little b, with anything for E, actually, it's little b, yes, and anything for E, you will be a brown lab. So the specific combination, it's like two separate, completely separate genes, but they work together to give you an outcome. I have a couple more examples of this. Another one is eye color. Eye color is multiple genes, and the interaction of those give us these various eye colors. Here's another example, the shape of the comb in poultry. So this is another example of epistasis. So particular combinations give us different things. So if we look at this, they're showing a particular cross, but here's the bottom line. There are basically four shapes to the comb. The four shapes are, and I'm going to get rid of this up here. So we're going to get rid of our pictures of our roosters. All right, so our four shapes that are possible are a walnut comb, a rose comb, a pea comb, and a single comb. The code for walnut is basically big R anything, big P anything. They're going to have a walnut comb. I'm getting this from looking at my F2s here. To have a rose comb, they need big R anything, but they have to be little p, A pea comb is little r, big P anything. And then a single comb is little. They have to be recessive for both. So they give you a scenario here. A walnut crossed with a rose yields a single. So we know that a single, remember, is little. That's really important information. So one of the parents is a walnut. So walnut is big R anything, big P anything. You say, well, okay, well, I don't know what the other letter is. Ah, but you do. Because even though walnut, that second letter, could be big or little, remember, their first child was little, 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 little. That means both of the parents have to be carrying the recessive gene. So our first parent that has the walnut has to be this. Big R, big P, little p. It also says the other one is rose. So if we look at the code for rose, rose can be big R anything, little p, little p. But we just said that the offspring, their first offspring, is little, 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 little. So again, we know that that second R has to be little. So this would be our original parents. Now we can solve this using the product rule. So we'll cross our R's. And the way you do this is actually a very complicated problem. So we have basically a 3/4 chance of being big R something. And we have a 1/4 chance of being little r, little r. And there's a reason I'm writing it this way, because you can't really determine what the offspring is going to look like yet with just the r's. And now we do the same thing with the p's. Big P, little p, with little p, little p. We're going to basically have a 1/2 chance of big P, little p, and a 1/2 chance of little p, little p. So now they want to know what kind of offspring you're going to get. Well, there are 4 possible types of offspring. Let's see how many of each type. So walnut, rose, pea, single. So the chance of walnut. So walnut, remember, I said was big R anything, big P anything. So the chances of having a big R are 3/4. The chances of having a big P are 1/2. And so 3/8. Rose. Rose, if you go back, is big R anything, little p, little p. So the chance of having at least one big R is 3/4. The chance of little p is 1/2. And so 3/8. Now pea comb. A pea comb is little r, big P anything. Well, our little r chances were 1/4. And our big P, little p, which is the only thing other option here, is 1/2. So that's 1/8. And then a single comb has to be all little letters. So there's a 1/4 chance of little r, a 1/2 chance of little p, and that's 1/8. And you'll notice, by the way, if you do these correctly, it should add up to 8/8. Like, it should account for all the possibilities. So this is a very hard problem to solve. If you can do this, I would say, man, you're in awesome shape as far as this chapter goes, because it would be unusual for you to have more than one problem like this on a test or on the AP exam even. All right, a different kind of trait is called polygenic inheritance. This is a little bit different. In polygenic inheritance, a trait varies along a continuum. In other words, what you usually see in polygenic inheritance, if you see a trait, and when you look at all the offspring that exist, you see a bell curve. For example, height's a really good example. Skin color is a good example. There are people that are really, really, really light all the way to really, really, really dark. That is going to imply that it should be polygenic inheritance. Now, what makes this different from epistasis that I just showed you? The difference is that in polygenic inheritance, this is for the color of skin. Polygenic inheritance, it's just the number of capital letters you have. We did this in the baby lab. You flipped and then you counted how many capital letters. The more capital letters, the darker you are. So if you have all capital letters, your skin would be very, very, very dark. If you have all lowercase letters, you would be very, very, very light. But the actual combination doesn't matter. In other words, you could be big A, big B, big C, little c. You would be the same color as big A, big B, little c, because both of these have three capital letters. So in polygenic, the actual letters don't matter. The actual code, it's the number of dominant copies, because basically you have several genes that code for making more and more and more of the same thing, not different genes that make different things that then interact with each other. And here's a picture of that bell curve that you get. This would not be incomplete dominance, because in incomplete dominance there's still only 3 options. Like with the roses, all you could have were red, pink, white. That was it, just 3 colors. So if everybody was either this height, or this height, that could be incomplete dominance. But if you see a whole bell curve of heights, that incomplete dominance doesn't explain it. It needs to be polygenic. It must be controlled by multiple genes.